And that is actually not the case. Our genome is actually highly dynamic. We have telomeres at the ends of our chromosomes. They change in length with time. We acquire somatic mutations. Our mitochondrial DNA, within every single cell, we have hundreds of mitochondria, changes in terms of copy number. We have retroviruses. We have our B cells and T cells that are constantly rearranging. And the scientific focus of our group is really understanding how some of these changing features of our genome contribute to health and contribute to disease. So today I want to talk about two different projects, one focused on genomic discovery, the other on clinical translation, and sort of highlight how we are actually using AnVIL on Terra on a daily basis to get this kind of work done. So talking to colleagues yesterday, they said, well, yesterday we heard all about AnVIL, how great it is, but like how are people actually doing things? And we saw that in the afternoon sessions. So I wanted to actually focus in on how we actually are using AnVIL and have been for years to make science possible. It wouldn't be possible otherwise. So we had a PhD student who wanted to join our lab at Vanderbilt. They do about a month-long rotation where they get to see if we're kind people and whether we take them out to ice cream or not. Whether we make them work on the weekends and things like that. And Hannah Poisner had just seen this paper come out in Nature that said, here's a new method where we can take whole genome sequencing data, look at the basically coverage over the T cell receptor, and from that you can estimate what percentage of the blood is T cells. And she said, well, this is a cool tool. They had run it on a few thousand samples. Wouldn't it be really cool if we could actually just run this on like 110,000 genomes? And then we could understand the genetic architecture of T cells, which no one has ever been able to quantify before. So how did Hannah actually do this? Well, it's basically 5 steps. First, she made a workspace. That takes a few minutes. The hardest part is actually the billing, which thankfully at Vanderbilt we've solved over the last couple of years, and trying to get connected with NIH strides. Then she took this tool that was this code that was published in Nature about TCR quantification, turned it into a Docker that really didn't take too many lines of code. We decided to run it on all top med genomes at the time. And so, again, in cloud computing, as you guys know, we can leverage other access authorities. This was through dbGaP. We had access to these data sets already. So to add Hannah to be able to access this was a few more clicks. And then she was able to then run this workflow on about 110,000 genomes. This took her about a week. And just again, this audience, perhaps this is not a surprising concept, but when I give this talk elsewhere, to download 110,000 genomes, this was about 3 petabytes of data, would have taken her a year just to download the data. And it would have cost her about $50,000 a month to store the data. And in our group, when we're routinely interrogating the actual sequence reads from these genomes to find the dynamic genome features, this is obviously not something we can afford to do, and really makes this kind of research uniquely possible. So she ran this. Again, we saw yesterday, workflows are really cool. We love workflows. And again, this is real. This is not a made-up example. This was a job submitted by Hannah on November 16th, 2021. About 8,000 of them worked, about 1,000 failed, and that's okay, because with one click, you can just launch them. Each one of these jobs cost about a penny, and so to do 110,000 cost us about $1,400, which, again, there's always a discussion when our faculty meetings about, isn't cloud expensive? And it's like, well, science is expensive, of course. But $1,400 is a whole lot less than buying the antibody that a different graduate student requested that same week. So no, it's not expensive. And then on AnVIL, she's able to make workbooks, as you guys saw yesterday in the demonstrations. and analyze the result and identify for the first time that the abundance of T cells in the blood does have a heritable basis. And she identified about 12 novel loci. And then there's one more step, which we still haven't figured out how to do on AnVIL, which is actually publish in peer-reviewed journals. We can certainly publish the workspaces, publish the workflows, and that doing the whole project took about a month, and then publishing it took another like 2-3 years. But that's how science works sometimes. And so this was really cool. And then meanwhile, the same group that had made the first tool came out with another tool. And this was a paper published in February of this year. And so Hannah said, oh, look, there's a newer method. Why not, this new method can do T cells and B cells. Let's just do it again. And so again, this method came out about 6 months ago. Hannah finished this analysis about 4 months ago. And instead of 100,000 genomes, now she wanted to run it on 800,000 genomes. The genomes through all of us, through TopMed, through UK Biobank. And now she's found about 700 novel loci that affect T cell and B cell abundance. And this is all science, genomic discovery science, that can only be done because we have access to tools and platforms like AnVIL. So that's what we're doing in using AnVIL to empower genomic discovery. What about clinical translation? I'm going to tell you about this thing called clonal hematopoiesis, which has been a fascination of mine for the past almost 10 years now. So what is clonal hematopoiesis? You can think of clonal hematopoiesis as almost a pre-blood cancer. So with age, we acquire mutations, and especially in our blood stem cells, which are constantly dividing, it's not uncommon as we get older for these mutations to occur in a gene that gives that blood stem cell a competitive advantage. So then that precancerous cell can grow and divide a little bit faster than its neighboring cells. And with time, a subset of these individuals go on to develop blood cancer. Now, this idea of having clonality in your blood, or clonal hematopoiesis, can take many different forms. We heard a little bit yesterday about how this could affect sex chromosomes, such as loss of Y chromosome, copy number gains, and copy number losses. But what I'm going to focus on today is one form called CHIP, or clonal hematopoiesis of indeterminate potential. CHIP is basically caused by point mutations, or short insertions and deletions, in a subset of genes that cause myeloid cancer. So there's many different forms of clonal hematopoiesis. CHIP is one of them. And one of the things that we and others found about 10 years ago is that CHIP is basically hiding in plain sight. That if you look at large-scale genome sequencing projects, even ones that are only 20x or 30x in depth, if you focus in on the myeloid cancer driver genes, you can find evidence of clonal hematopoiesis. So this is a view from IGV, Integrated Genome Viewer. And what you see is that each gray bar is a next generation sequencing read. And you see three of these bars, three of these gray bars that have a 1 base deletion in them. When you have 3 reads that have this 1 base deletion out of about 20 or so, that suggests, or that's a signature of having a somatic mutation in 3 out of 20 or percent fraction of your blood. So we're able to use these kinds of signatures to find that in sequencing that was done for diabetes or Alzheimer's or some CCTG, pretty much any large-scale sequencing consortium, we're able to find this. Now, as you might imagine, next-generation sequencing is very error-prone, so we actually need to have better tools, or better algorithms computationally. So several years ago, we sort of formalized how do we do this well, and you can look at a workflow that looks sort of like this, and we use Mutec to call somatic mutations, and we annotate and we filter, and then we filter some more, and then we filter some more. And then, eventually, after lots of filtering, we come up with a list of mutations that we think represent these somatic mutations. Um, we've described this in terms of a flow diagram like this, but then more importantly, we built this as a WDL. This WDL pipeline lives on AnVIL, lives on Terra, has been deployed in other platforms now, like DNA Nexus and others. And our lab has personally run this on about 1.5 million genomes. We've personally run this on about 250,000 targeted sequencing samples. And this flow diagram basically has now been accepted by the larger bioinformatics community as one of the standard ways to identify CHIP. And so when we talk about what is the power of having these platforms like AnVIL to do large scale science, it's also to do reproducible science, as we heard a lot about yesterday. And that when people ask us how do we do it, we say easy, just log on and download our WDL and run it, and you're sort of off to the races. So we do this at large scale. This is data from all of us. What we find is pretty remarkable. Almost 10% of people in their 70s and above have these somatic mutations in blood cancer genes floating around in their blood. And what's fascinating to me from a biology perspective is that the drivers are not your sort of typical cancer drivers. They're not, there's a small number that are signaling, cascades or DNA damage repair genes. Most of them are actually changing your epigenome. Genes like DNMT3A and TET2 that put ethyl groups on or take methyl groups off CPGs. And so, again, a large part of the discovery work in our lab is also focused on trying to understand how is it that just by changing the epigenome, you can completely reprogram blood stem cells. This has become not just a sort of research curiosity, which is how I might describe it when we started doing this, but actually is now a real clinical headache because the same somatic mutations in the blood are found on solid tumor genetic testing. And so every week when our cancer center colleagues routinely send panels to detect somatic mutations in tumor, they actually find the somatic mutations in the blood, and then they refer them to our clinic that I helped start here at Vanderbilt called our CHIP clinic. And the reason they do that is because CHIP has been linked through leveraging the kinds of data sets that I was just telling you about, not just to blood cancer, but actually to a very large number of inflammatory diseases, and ranging from heart disease to stroke and liver disease, kidney disease, gout, and so forth. What we found across all of these large-scale biobank studies is that larger CHIP clones have worse outcomes. So if you look in the UK Biobank, at the variant allele fraction, which is the percentage of blood that has the mutation, if you have a small clone, makes up less than perhaps 5% of your blood, not such a big deal. But if you have a clone that's all of a sudden taken over more than 20% of your blood, now your risk of dying is almost twofold. Your risk of developing a blood cancer increases by almost 30-fold. And so we've been very interested in understanding, not just the disease consequences, but why do some clones grow? Because if bigger clones have worse outcomes, then we hypothesize that maybe faster growing clones are worse than slower growing clones. Which led to this question of, well, that's interesting, but how do we actually measure CHIP growth rate? And so on one hand, you can think about this hard and say, measuring growth rate is pretty easy. You just need 2 different blood samples. That if you're detecting this in the blood, just need one sample from when someone's 60, one sample maybe 20 years later, and you look and see, okay, over 10 years, the clone has grown by 10%, 1% per year. Not such hard math. The challenge was when we looked around the globe at all of our global biobanks, almost every single one of the biobanks just has one blood sample from a person, because all of these biobanks thought very hard about this, and they said, well, your genome doesn't change, so why is it that you would need more than one blood sample? And so I was fortunate to be here at Vanderbilt where that was generally true. Most of our blood samples, we do only have a single time point for. But actually, I learned that 10% of BioView, we do have a second blood sample because BioView is this sort of depletable resource. And so once the amount of DNA in the freezer gets below a certain amount, they get a second sample so that they will always be able to renew the amount of blood they have. And that threshold for how much DNA should be left in the freezer before they get a new sample was set 20 years ago when you needed micrograms of DNA. And now that our assays to detect clonal hematopoiesis need nanograms of DNA, there are a lot of these, quote, depleted samples that actually were still in the freezer that we were able to use. So we did a study where we said, okay, let's start sequencing people at more than one time point to understand, how are their clones growing and changing. We looked at about 3,000 people, 700 of whom had CHIP. These were older individuals, and looked, both at first time point, second time point, how are their clones growing and changing. And what we saw was actually pretty surprising to us. We had this mental model going into this project that the clones, once they exist, they just grow and grow and grow and grow. And if you live long enough, they turn into blood cancer. And that's actually not at all what we saw. This is one of the genes. This is ASXL1. And each line represents one person. And the red lines show the clones growing. And the gray lines show the clones staying about the same size. And the blue lines actually show the clones shrinking or starting to go away. We said, oh, that's interesting. Is that true generally? And yes, that's actually true across almost every CHIP gene. There's some of the clones that are growing faster than others, some that are stagnating. And except for just a handful of genes, almost all have some small number of clones that seem to be going away. So this was really interesting and also really puzzling because we thought, well, if we can identify why or how these clones are shrinking or going away, we'll have much better tools for telling patients when they come into our CHIP clinic, you need to be worried, you don't need to be worried. And so first we're focused on what about the driver gene? And certainly the driver gene is a big determinant of how fast the clone was growing. Some genes grow faster than others, but there's a huge heterogeneity, even across different genes. So one of the cool things about BioView is that the DNA samples are linked to the entire electronic health record in a de-identified way. And we said, well, I wonder whether there's some medicines that people are taking that maybe are causing the clones to grow or shrink at different rates. It's sort of a drug repurposing study of sorts. And we found again, this is all somewhat underpowered, I should say, very underpowered, but we found a couple of interesting candidates. One, the top hit of which was this thing called colchicine, which is an anti-inflammatory medicine that people routinely take for gout, now has an indication for cardiovascular disease. And so we made the observation that colchicine seems to cause clones to grow less, to grow more slowly. And then, so we put this out there about maybe 6 months ago. And then just last week, a group of investigators actually reanalyzed a colchicine clinical trial and found the exact same thing, that among certain CHIP genes, particularly TET2, patients who were randomly started on colchicine compared to placebo had their clones grow much less quickly than individuals who were on placebo. And I think there's another study that I've heard has also replicated this in a different randomized trial. And so for us, again, this is really exciting because we can use data resources, platforms like AnVIL, which again is where we do all of this analysis, to identify clinical hypotheses that we can then take forward. So how do we actually do that? Well, in order to actually move these drugs towards a clinical trial, we need to identify which of the patients who have clonal hematopoiesis are most at risk for progression. We work closely with several investigators to do this, where now we have a risk score, and more importantly, we have a website. So when patients come into the clinic, we can, doctors are not so good at calculating things, but they're not so bad at pressing buttons. And so we are able to, come up with these estimates of based on a very small number of clinical features that are in front of the doctor, who's at highest risk of dying. And in fact, 90% of patients with CHIP are at no risk of anything. And so trying to be able to tell the patient who comes in very worried about this that you're actually going to be fine, and also being able to risk stratify the very small fraction of people who are in a much higher risk category is helpful. We've extended this again using the Terra and AnVIL platform to look at progression to something called cytopenia, which is an abnormal blood count. And we've found that, again, in a very small subset of people who are particularly high risk, their rates of progression from just having this clone in their blood to having abnormal blood counts, which puts you at high risk of infection and other complications, is actually quite high, that we can identify a small group that might actually benefit. And we also use AnVIL to then try and, in our clinical cohorts at Vanderbilt that we use to study clonal hematopoiesis, to actually find these patients. So when I came to Vanderbilt in 2020, we started recruiting patients who came into our CHIP clinic, recruiting others who are at risk for CHIP. We have several hundred patients enrolled this way, and we follow these patients every 3 to 6 months and get new blood draws because we've learned the hard way that their genomes do change over time, and we're very interested in this. We leverage some really great work done by Paul Harris and his team with REDCap to pull in data from the EHR. We store all of our sequencing data on our version of AnVIL, and routinely test these samples for CHIP and other things. And then we actually return the results of the analysis that we get off of AnVIL to our research participants. We refer them for CLIA clinical testing when appropriate. And we use this study to actually do, as the data that is being crunched on AnVIL is actually feeding real patients into real clinical trials. We were impressed by this. We've launched a bigger one that's more of a screening study. It does the same thing with some additional bells and whistles. Again, sequencing data stored in AnVIL. And then at Vanderbilt, we have 4 different clonal hematopoiesis clinical trials that we are actively enrolling patients in. And where do these patients come from? They come from these resources, these studies that we built on AnVIL. So I've told you a little bit about genomic discovery, how we use AnVIL, a little bit about clinical translation, and how we're actually trying to change patient care using resources on this platform. And I wanted to spend the last 3 minutes or so reflecting on my personal opinion. I don't speak for the consortium in any way, about sort of some of the challenges and some of the opportunities we have on the road ahead to use platforms like AnVIL to their fullest potential as we try and change genomic medicine. I'd say one opportunity that this consortium has long recognized is trying to think about how do we embrace this sort of multi-cloud future. So this was an analysis that I did in collaboration with all of us in the UK Biobank, where we tried to do a very simple analysis, a simple GWAS across 2 platforms, doing sort of traditional meta-analysis. And then we said, well, what if we can just stage the data in Community Terra, access it through the All of Us workbench, and do it in a pooled way instead of a meta-analysis. And not only is it half the number of steps, takes half as long, costs half as much, you actually get more results and better results when you do it in a pooled way. And so thinking about how do we federate data across clouds, how do we access data across clouds, is something that I think is a huge opportunity. Another opportunity that we're thinking a lot about at Vanderbilt is how do we actually repurpose the genomic data that's being generated in the course of clinical care for research, and how do we make it accessible, whether to the larger world's research community through AnVIL or through our local research community. This is a paper published by colleague Lisa Bastarash in Biomedical Informatics about just over the past 20 years, how have genetic tests grown at Vanderbilt? And looking across the entire electronic health record, you can see that in the past few years, we're now actually generating quite a number of genetic panel tests. We're doing quite a number of exomes every year just as part of clinical care. How can we take that data that's currently living in our clinical laboratories and move it into the research space? And this is something that our corner of the AnVIL consortium has been thinking a lot about, not just at Vanderbilt, but across many different clinical labs. How can we use the electronic health records to identify patients who should get this genetic testing? How do we, once we have the genetic testing, actually integrate it into routine clinical care? And these are things that with our Genomic Learning Health System Network, which was launched a little bit more than a year ago by NHGRI in collaboration with National Cancer Institute, we're going to try and do these things. How can we take data, real world electronic health record data from many different systems, stage it in such a way that the world can learn from that? And I think this is something that our network is really excited to collaborate with the AnVIL team on in the coming year as we start to actually launch our first network project. So with that, I wanted to thank everyone in my lab. We dressed up as a karyotype for Halloween. Many collaborators across Vanderbilt who contributed to the work. Obviously could not be done without funding from NHGRI and in collaboration with other institutes across NIH. So thank you.
I think we have time for maybe 1 or 2 questions.
If you don't mind repeating the question for people online, thank you.
So thank you for a wonderful talk. It's really open, from because longitudinal data set is like statistically driven to workers, and we finally have the largest sample size in all of us. But my question is very technical. You touch on federation. So when you have analysis pooling from all of us, and then other resources, are you done it currently in all of us workbench environment, or you are able to actually from your AnVIL environment and access all of us?
Yeah, great question. So let me rephrase. The question is, Alex, you showed us some smoke and mirrors about how you could do things across UK Biobank and all of us. What platform did you actually use to do it? So we did this in 2023. And at the time, UK Biobank data was allowed to exist outside of their trusted research environment. And so we actually had a copy that a large number of groups at Broad had used that was staged on Community Terra, basically Terra on AnVIL, or AnVIL on Terra. I can never remember which way we're supposed to say it. And all of us had their security boundary. We had applied for basically an exception to be able to reach outside of the all of us security boundary to pull data into the environment. So the actual analysis was done inside the all of us researcher workbench, which is also basically just Terra, where was at the time is starting to evolve a little bit, as you heard yesterday from Paul. And so the analysis was done when we tried to pull them together. The joint analysis was done within all of us workbench. I think all of us, as exemplified by the work shown yesterday with the combined AnVIL all of us imputation server, is increasingly excited to partner beyond the network limitations of all of us. And so I think that there is the work that was done in 2023. It continues to be sort of a use case for them of, oh, this is something that we really, there's a scientific reason that we need to get there. And again, how we get there, how quickly we get there, that I have no idea about. But I think the ultimate goal across all of, there's no reason if the security perimeters and security policies are just as robust and strong, and the identity management can be figured out. Again, from my personal opinion, not speaking on behalf of all of us or AnVIL, I don't see any reason why in the future it has to be launched from within the all of us space. I would love to be able to see federated analyses launched from from any of the clouds that are coming together for this work. You can shout and I'll repeat
Hello.
it. Oh, now it's on.
Okay, well, just for the people on Zoom. I was wondering, what are the patient reactions that you got from the China cohort? Are they getting excited about this research?
Yeah, great question about what are we hearing from patients when we return these results to them. I think patients are really delighted that we've come up with a way of sort of further risk stratifying. Because when we started doing this maybe 5 years ago, everyone said, "Oh no, you found a cancer mutation in my blood. I'm going to get blood cancer and die." And now being able to tell them, actually, we did find a cancer-causing mutation in your blood, but you have no greater risk of developing blood cancer than anybody else, based on some of these risk scores, and that's been very, very helpful as wearing my doctor's hat. Thank you.
One last one. Great privilege here. So first of all, thank you for an outstanding presentation, and also thank you for all of the clinical work. I think this is why many of us here get really excited about AnVIL, is translating this basic science into like real patient lives. So thank you for that. You brought up the sort of realities of multi-cloud. I'm with you, and we are trying to build technologies that will help facilitate that. I'm wondering, you have our full attention. Is there anything within AnVIL, Terra, and beyond specifically that we can help you with? Is there a feature or some other functionality that you would love to have? I'm very curious.
Yeah, we've been great questions. So thinking about what is my group's biggest pain point? I think that there's still places that we can do better about some of the federated data access. That some of the linkages into dbGaP are sometimes break for reasons that have nothing to do with you guys, and then it takes a couple of days to figure out why are they broken. And so I think there's continues to be opportunity to try and get some of the authentication pieces to work a little better, which is again a very small ask maybe, or a very big ask depending on which part of the consortium you're looking at. But I think whenever people are coming to my office complaining to me, it's usually around that interface between data that lives in the platform that we've uploaded and data that they're trying to access elsewhere. I think what people love is the workflow management. I think that has saved many, many, many grad student years of life. Yeah, but again, we continue to be huge fans, huge evangelists for using this platform across every new lab that sets up at Vanderbilt, where you tell them, don't bother trying to go to a local HPC cluster. It's going to be down, and you will always have AnVIL.
Thank you so much. Thank you.